Tom, thanks a lot. Boy, I have to say, I just listened to the interview you did with Peter Attia.
Oh, yeah.
And I think that came off pretty well. I mean, it's a small seminar in itself.
Yeah, well, he was here in my office, and he spent quite a while. And I said to him he could ask me whatever he wants to ask. And he was, as you heard, I think he asked me quite a bit of questions. And then his colleague, Bob Kaplan, was here in the office to collect the references and in support of what I was saying.
Right. You know, in part, I feel like I'm asking you to repeat so much of your information, but it's so fascinating. And every time I thought I had a handle on what you were talking about, I realized it was a deeper level. And I'm going to jump ahead, but we'll go back to the beginning. Is like the whole substrate level phosphorylation was something that I was unaware of. I thought I knew Warberg and everything else, but let's get into that later because that's a big key to of all this. But some of the personal questions I have is like, you were a Vietnam vet.
That's right.
And then how did you go from there to, gosh, I think I'm interested in biochemistry?
Yeah. Well, I had done biology before I went into the Army. And, you know, I was very interested in genetics, but at that time in 1968, everybody was being drafted.
Yeah.
So it wasn't the plan. No one could really make plans to do anything.
Right.
You know, so I just decided to join the Officer Corps in the Field Artillery at Fort Sill, Oklahoma. And then I spent about a year in Germany and then a year in Vietnam with the 1st Cavalry Division. So I spent almost my whole time in the jungle. And maybe 60% of the time in the jungle and 40% of the time on the fire base. So it was kind of split between jungle and fire base. You know, neither one was what we would call a comfortable existence.
So was your perspective like, I don't know if life's going to last another 10 minutes, but I might as well apply to, graduate work and see what I'm.
Yeah. So I had already thought about applying before I had gone into the military. And then, as I said in Travis's book, I actually applied to graduate school while in the jungle and the paper was full of mud and stuff. And I just never thought anybody would accept an application like that. But they did. I went to Illinois State University, got a degree in genetics, master's degree. And then I went to the University of Illinois and got a Ph.D. in genetics and biochemistry.
At Urbana, right?
At Urbana, yeah. Champaign-Urbana. And then I did a lot of work on lipid biochemistry and mammalian mouse genetics. And I just followed that with a postdoc at Yale. And then I got on the faculty in the neurology department at Yale. It was a clinical department of neurology.
Right.
So, it was mostly epilepsy work. I wasn't doing any, at first, I wasn't doing any cancer research there at all. But then we started to look at gangliosides in tumors.
Mm-hmm.
And I started to get interested in tumors in the mouse. And then we started to, it was always a side project. I had a couple of NIH grants over the years to support the work, but most of the work I was doing was in epilepsy.
Right.
But the cancer stuff was on angiogenesis, which is blood vessels in the cancer cells,
Mm-hmm. Mm-hmm.
And people thought that was hot, gangliosides control angiogenesis, and it was academic stuff. I couldn't see off the bat how any of it would be applied to any person in the clinic. And I would say 90% of all cancer research is done with no interest, not an interest, with no connection to what goes on in the clinic.
You say that because somebody just needs to come up with another idea, get it funded, and move on, but not have a.
Yeah, your job in cancer research is to sustain your research program. It has nothing to do with keeping people alive in a cancer clinic. I mean, that's a fact.
Yeah.
They go under the umbrella. It's cancer research because you're studying some tumor cells in culture on the ass of a mouse, but it's just it's not like, oh, this therapy is definitely going to have an impact. But it's funny because everybody, I would say, 80% of the papers that are written in cancer research, this finding could have some significance in the clinic, right? And that's the end of it.
It is.
This could have some value in the clinic. This might do this. This might do that. But nobody's doing anything.
Right.
So we have 1,600 people a day dying for things that might help them.
I agree. Well, now I understand why they say that. Let me go back because a couple things. It's really interesting. You were working on epilepsy. which you would have
Oh,
thought
yeah.
would have been automatic to the ketogenic diet, but you took kind of a different route and you came back to impart the ketogenic diet as part of being the metabolic therapy. Would that be
Yeah,
correct?
well, we started with calorie restriction. And because it's well known, documented in the literature, that just cutting calories can significantly reduce tumors. And we found that we and others
Yep.
had shown that this is linked to angiogenesis. So if you want to stop angiogenesis, reduce calories. And then we found, and others found, that one of the mechanisms for calorie restriction is reduced inflammation. So everybody knows that cancer cells and tissues are inflamed with the growing tumor. Wow. So calorie restriction reduces inflammation, but we had to show that it works through the NF-kappa B signaling system. So each of these observations had to be linked to a series of signaling cascades. AKT signaling for VEGF, and people, the guys in the cancer field love mTOR, AKT, they like all that stuff. So you got to do something that, that's like, you got mTOR. Wow, this is the most important thing. So you do this stuff. Only you need to get money, right? If you say you're not going to study AKT signaling, it's very hard to get any money, or VEGF, or what's the molecular mechanism.
Right.
We spend so much time in the field looking for molecular mechanisms for things that may have no relevance to the clinic.
Yeah.
So there's two fields of cancer research. One is just pure basic stuff, and the other is clinically applicable. So you think that the basic research should set the platform for why something should be tried in the clinic. And that, I think, is should be the purpose of cancer research, is to say this work that I'm doing will have a direct application to what's going on in the clinic.
Right.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
And I think that's important. So, we were doing all this on mice, and we also developed some of the best animal models for the disease in my laboratory over the years. I worked indirectly with Harry Zimmerman. Harry Zimmerman was the physician who started the first department of neuropathology in the United States at Yale University back in, I think it was the late 20s, early 30s. And he was still alive when I got into the field.
Was he one of your mentors, or he just happened to be there?
He was at Mount Sinai in New York City. But I had a lot of phone conversations and letters that I had communicated, because he was the guy who started doing the first brain tumor work, one of the first, by putting chemicals into the mouse's brain and developing brain tumors, and then classifying all these tumors and showing how similar they were histologically to human tumors. But in my book, I think in chapter 3, I exposed the fallacy of the ambiguity of how anybody looks at brain tumors. It's a real mess. No, everybody has a different opinion, and it's what it is group consensus. If you allow neuropathologists to make a decision on their own, it's often very different from their colleague that would make an independent decision on his own. So they all come together as a group, and so it's a consensus thing. So when you go around the field, all this consensus, and then you give these people individual tumors by themselves to ask, and everybody has a different opinion about what it is. So it became clear to me that these guys don't know anything about what they're doing. Who cares? Classification of the tumor. And does it make any difference what you're going to do to the patient after you classify the tumor?
Right.
No, you're still going to cut and irradiate the guy. So what difference does it make what kind of 12? It's this. And then if you go in and cut the tumors out too prematurely, you cause a low grade to become a high grade. And they don't like to talk about that.
Mm-hmm.
But, a good 25 to 30% of all glioblastomas started as low-grade tumors.
Mm-hmm. Mm-hmm.
It's actually a terrible situation. I'm going to write a paper on trying to change the standard of care, because what we're doing to these brain cancer patients is
I agree.
terrible.
I agree. Absolutely. Before I get the horse in front of the cart, I wanted to go back when you were studying lipids, and you were, you came across a company that asked you to do some research.
Yeah.
And then you did, and you realized it wasn't the drug at all. So,
no,
but that's a great
calorie restriction.
That's right.
So we wrote a chapter in a book on indirect calorie restriction. A couple of people had mentioned this. Actually, Albert Tannenbaum a long time ago had mentioned this about drugs, that if you give a drug to a mouse and it makes them sick, mice don't vomit. They just stop eating. Mm-hmm. And what happens is, if the mouse stops eating, the tumors start to shrink. So if you take the drug out and just restrict the food intake to the same level as the drug, the tumor shrinks to the same level. Mm-hmm. Many times, not all the time.
And that's exactly what you did in that particular, right?
Yeah. So and besides, the drug targeted gangliosides. It wasn't like the drug wasn't. So it was, at the first, it looked very interesting. The drug that we used blocked ganglioside biosynthesis, but also shrunk the tumor. So it was the company that made the drug was very excited, because they thought that it was the reduction of the gangliosides that was responsible for the shrinkage of the tumor. But we then later on found out that the drug, the restriction of the gangliosides had nothing to do with the shrinkage of the tumor. It was the food intake. So we did some control experiments, and I can't remember the details. I'd have to go back and look, but it wasn't the gangliosides. Got it. So and that got the drug company upset, because they thought they had a thing there. It turned out to be, again, indirect calorie restriction.
They want their money back?
Yeah. Well, that's actually what happened to what's the woman's name? Yeah, she's kind of famous. She worked with a drug company, and they spilled the beans in advance. She pulled her stocks out of the drug company. But I look back at that drug. Martha, was it Martha Stewart? Yeah, it might have been.
It was Martha. Yeah, it was. She spent a year in jail. Right.
So I went back and looked at that drug that got her into all this trouble, or that company, I guess. And in my mind, it was working through calorie restriction. Wow. So when they give the drug to the mice, the tumor shrunk, but their body weights also shrunk comparatively. Mm-hmm. So it was clearly, in my mind, the major effect of whatever that drug was, independent of stock investments. Right. It was working through calorie restriction, but it had bigger implications only because of all the other riffraff that went around associated with it. But I went back and looked at the drug, and I said, damn, that drug that's the same thing I was looking at, something that was calorie restriction. Mm-hmm. So then you say to yourself, how many of these drugs are in fact working in part through indirect calorie restriction? Mm-hmm. And how many people who receive these drugs, who stop eating and get vomit and all this other stuff, is working through indirect calorie restriction? So it's very hard to design an experiment to control for fatigue, diarrhea, vomiting, and nausea. So but you have to do that in order to identify what the therapeutic benefit is. And generally, it's a combination of both. The drug is targeting and killing some of the tumor cells, but also at the same time causing the host to restrict intake of calories for a variety of reasons. Mm-hmm. And together, you get some sort of a therapeutic benefit. So, and that's the whole field has never been evaluated clearly like that. Right. Albert Tannenbaum, in my mind, and some of the other early investigators, they were the guys that did a lot of these kinds of studies. And it was very clear back in the 1940s and 50s, that you really have to be careful when you do these kinds of studies.
So, Tom, when you got into, the calorie-restricted ketogenic diet, there's calorie-restricted, take out the protein, take out the fat. So how was it still epilepsy in the back of your mind, realizing, ketosis is part of this? When did you switch to, it's now, one of the cornerstones of the overall metabolic therapy that you're working on, but it didn't necessarily, you can go from calorie restriction to calorie restriction ketogenic diet. How did that?
Well, we felt that to tell human beings that the best way, a good way to manage your cancer is stop eating for a month. Well, they're going to look at you like you're some nut, right? So, but that works. Actually, I've got papers. I've got people calling me from different countries who actually done that to people. 30 days of just water only. It's unbelible.
I would love to read those. That is such a great topic. Absolutely.
Yeah. Just, it works. And humans just have to do it a longer time, 14, 21 days, these kinds. And people say, oh, I'll be dead. No, you won't. I talked to George Cahill at this before he died. He was the head of the Joslin Diabetes Center here in Boston.
Yeah.
And George and I would go for hours talking about all these kinds of things. And he would tell me stuff that, you can never do today in today's medicine that he did to people back in the 40s and 50s. But
and 60s.
Yeah, and 60s. So he did a lot of that stuff. But no, human. So it's not practical. And when we found out in the mouse that when we restrict 40% calories, because they have a basal metabolic rate that's 6 to 7 times higher than ours, 40% calorie restriction to them is like a water-only therapeutic fast to us. So, and then we did all the biochemical parameter measurements, and their blood sugars and ketones shifted in the same ways that a person water-only fasting would do.
Okay.
As a matter of fact, when humans do water-only fasting, you can bring your blood sugars down lower than the mouse, and ketones much higher than a mouse can get.
Wow.
So humans are supremely designed to go long periods without eating.
To starve. Yep.
Yeah. A mouse will die in 5 to 6 days without just water with no food.
Right.
Humans are just getting started at 5 and 6 days. The body is just starting to ramp up.
Right.
So we realized that you couldn't be seriously recognized if you're going to tell people to stop eating for 30 days or something. So having worked in the field of epilepsy as long as I did, knowing how we stopped epileptic seizures with ketogenic diets, and we published some really beautiful papers in the field of epilepsy, where we were able to identify glucose as the provocative variable. And to this day, no one's really sure how the ketones are actually blocking the seizures, what role they play. But clearly, if the glucose is high, you can't stop seizures. And then I had long talks with the late John Freeman. John was the person who held on to the ketogenic diet
at Johns Hopkins.
Absolutely. At Johns Hopkins, yeah. He was a friend of mine.
Yeah.
Yes. So John and I would have very long conversations at the epilepsy meetings, discussing all of the variables. And he told me one time that in his practice, he would find every now and then you'd get a kid that liked the ketogenic diet, and they would eat a lot of it. And he said those kids we could never manage seizures in those kids. The kids that would eat a lot of ketogenic diet, because most kids don't like it, and they eat only as much as they have to. And it was actually, and human beings are a little different than mice. We have an anti-appetite effect from fat. It triggers the hormone cholecystokinin, which impacts on the 10th cranial nerve, the vagus nerve, to shut down appetite. Yeah. The mice don't have that. They do have it, but its effect is not as robust as you see in humans.
Got it.
So kids don't usually like to eat ketogenic diets. So we have to make sure they get enough so that their body weights continue on a trajectory that would be appropriate, age appropriate trajectory. Right. Otherwise, we're going to cause them to have developmental delay.
Right.
But if kids eat, a buttload of ketogenic diet, like this kid was pouring oil on ice cream, and he was like keto ice cream with oil on top. He loved oil. It was like oil everywhere. And we couldn't stop. John told me we got a couple of these kids every now and then, and they just get into this diet, and we can't control their seizures, so we have to make sure it's always restricted. So most humans will restrict it, but so in the mouse we must restrict.
Got it.
Now, some mice, it's all dietary dependent, because I have some strains of mice that if you transition them from a standard carb diet, mouse chow diet, to a keto, they would starve to death. They would not eat the ketogenic.
Wow.
So what we did is we put them on a water-only fast for 18 hours, which is like 1 week in a human, almost 5 or 6 days, so that they're really, really hungry. And then we'd give them the diet, and they would eat it. But we would still have to restrict it so that we would have a comparable reduction in body weight. We did a lot of these kinds of physiological studies and found out that it's the body weight that's the most critical variable. So when you compare animals under different dietary conditions, you must make sure that their body weights are exactly the same.
That sounds incredible to do, really hard to do. So you had the controls you had to measure every day and saying, well, we got to match them to the...
Yeah. Yes, absolutely.
Wow.
Absolutely. So it's a lot. So labor intensive. That's why our research is not cheap.
Yeah.
We have to pay a lot of people to work on these. It's like having a clinic with all your patients being mice. So, each mouse is in his own cage.
Yeah.
We measure his body weight every other day. We measure how much food he ate, how much water he drinks, his body weight, and then we treat him. And we got to make sure that, his body weight stays the same, so we don't run into inadvertent calorie restriction. And then if you're doing brain cancer research, which is very fascinating, they get a massive binge eating appetite. When you injure the brain, you cause a massive increase in basal metabolic rate. It's unbelievable.
Mm-hmm.
So the mice will eat sometimes 30 to 40% more food than they would normally eat, but for the fact that you now put a tumor in their brain. And we did sham controls where we would just put the trocar into the brain to damage the neural tissue, and then sew them up. And you watch about 3 days after the surgery, they would start eating like crazy. It was unbelievable.
Wow.
So apparently they needed the energy to correct the neural tissue. So we had to balance all of these different factors together when we were doing these kinds of things. So you have to keep, otherwise you cannot interpret the data. It becomes extremely difficult for data interpretation. If you're looking at molecular mechanisms of angiogenesis and inflammation, and you're not controlling the physiological variables, then you'll be misinterpreting the data that you collect.
Right.
So
right.
it took us 15 years of trial and error kind of experiments to do and learn about the physiology of the mouse in relationship to the physiology of the human, to do these comparative, so that when we were go into the clinic, we would be able to make recommendations as best we could that might be more appropriate for the human species than for the mouse itself.
Okay.
So we did a lot of 15 years and 20 years of basic research.
I have no doubt. I mean, just any of your studies sort of leave me in awe. But let me just sort of put it in my words. Were you on the path of realizing that calorie restriction was the key, not so much the ketogenic diet? It was the dropping the glucose and the fact that there was ketones that feed normal cells. Well, that's a good thing, but it's really the lack of the glucose that is the lowering of the glucose is the bigger deal here. Is that? Yeah,
well, it's a combination of both. When we worked with epilepsy, as I said, you couldn't manage the seizures unless the blood sugar was down.
Got it.
And then, of course, what did the ketones do? Well, ketones are known to be neuroprotective. We know a vast amount of information from Hans Krebs, from the Krebs cycle, and his last graduate student, Richard Veitch, from the NIH. Right. I mean, these guys had looked at the role of ketones, and Veitch showed that they reduce reactive oxygen species. They increase the delta G prime of ATP hydrolysis. So they do a lot of good things. But the cancer cells, from the Warberg theory, they need fuel. They need fermentable fuels. So the ketogenic diet is simply taking away or reducing a key fuel, while the ketone elevation is simply allowing normal cells to function in the absence of glucose, and protecting the normal cells. So not that the ketones are, we're not sure if they're responsible for killing tumor cells by elevating ketones. We're just using the elevated ketones to allow us to marginalize the tumor cells, which are dependent on the glucose. So it's an elegant internal system that allows the normal cells of the body to function appropriately, while the tumor cells are struggling to survive. So the ketone elevation is designed primarily for two reasons. Number one, to protect normal cells, and number two, to allow us to push blood sugars down to levels that people would say would be life-threatening. But we know from the work of humans, Denkler or somebody, where they fasted for 30 days, I think it was 20 or 30 days, 20 days, water-only humans, and then gave them large dose of insulin. All right, if you took that same dose of insulin and gave it to anybody, you'd kill them. So they got the blood sugars down on some of those people to 0.5 millimolar or 9 milligram per deciliter. Now, as a physician, this guy should be dead. No, he wasn't dead. There wasn't any hypoglycemic associated with him, but his millimolar ketone was 4 to 6 millimolars in the blood. So they completely protected his brain from a level of sugar that was almost unmeasurable. So that told us clearly that if we want to kill cancer cells that are dependent on glucose, we just have to transition them to therapeutic ketosis and then hammer those glucose levels down. And the other beautiful, which we showed in our mouse models, is in order to get, glucose into cells, you have to have receptors. There's the gluc transporters,
Right.
and there's a slug of these glucose transporters. Gluc1 is the major one.
Right.
Well, cancer cells are garnished with these glucose transporters, so they're taking in as much glucose as they can get. But when you take glucose out of the microenvironment, the normal cells of the brain and other areas upregulate those transporters because they want the glucose too.
Right. So now you have a competitor.
Yeah. Now you're using your normal cells as a direct competitor against the tumor cells. So this puts even more pressure on the tumor cells.
Nice.
So if you can, so therapeutic ketosis, which essentially involves low blood sugar and elevated ketones, puts tremendous pressure on tumor cells that require glucose for survival.
Right.
So that's why the ketogenic diet done appropriately is such a powerful therapy for targeting all kinds of cancers. One of the interfering problems is the standard of care. It seems like we always have to treat somebody with some toxic chemical because they can't understand that you might get the same level of therapy if you didn't use the toxic chemical.
They're a little vested in that. Yes.
Yes. Well, I'm saying, I think in some cases these toxic chemicals interfere with the elegance and the beauty of the ketogenic diet, just so that we can, say that we treated this patient with something.
Right.
But, why don't they treat him with the metabolic therapy rather than the toxic chemicals?
I'm with you on that for sure. So I want to back to the question of, so now I have the ketones and the glucose, and we can even go back to Warburg. Do you have, and I know you have the glucose ketone index and better than one, but and that's a 1-to-1 millimole to millimole. Did you sort of, how did you come up with that number? Saying, well, at least we need that. Because I wanted it, now one little paragraph too, we had there's a group of us, we went through a group fast about a month ago, and I had everybody keep a spreadsheet. And so we calculated that. And for a number of us, I was in the 3s, but then on day 3, it snapped to 1. And then I did a 7-day fast below that. I was, after day 3, I was under 1, and my lowest was 4.5 or something, 0.45. But it was interesting that was the pivot point. So in one way, how did you come up with that? And
well, we came up with it from a person that we worked with, Trudy Dupont, who unfortunately passed away.
Okay.
She had a diffuse infiltrating brainstem glioma. She lived 7 years with this. So we kept her, we think we kept her alive for a much longer period. She took no radiation or chemo. But when we initially said you should keep your blood sugar at 65 and your ketones at about 3 to 5 or 3 to 4, somewhere in that area, millimolar, of course. So she was religious in every day measuring independently her glucose and ketones and charting and plotting and giving us the numbers. And we plotted them out over a period of 2 years, if you can believe this.
Wow.
And the variations were huge. She got into a fight. She had a handicapped parking spot. Oh, she lived in Nice, France. She was an American lawyer, graduated from the University of Wisconsin. Tremendous woman, run marathons and did all this. Very attractive woman. Really, really athletic. And then she was diagnosed with this kind of tumor, but she lived in Nice, France, and she had a handicapped parking spot. She had a little bit of a cane. She walked initially with a little bit of a limp, but then a cane. Anyway, she got into an argument with the neighbor who took her spot. And I know she went upstairs and measured her blood sugar, and it was like 150 milligrams per deciliter. And she had been keeping it really low. And she emailed me and she said, every time in the morning it gets really high. And then when I get into a fight with my neighbor, and now I know my tumor's going to grow faster. And I said, what's your ketones? And she said, no, they're still pretty steady at about 4.5 to 5 millimolar.
Wow.
So I said to my student, Josh Meidenbauer, because we were the ones who published the paper.
That's right. You made the app in essence, right?
Yeah. So I said to Josh, I said, this whole thing about the glucose is too capricious. It can vary from so many different kinds of things in the environment. An argument to the morning effect, all these kinds of things. But the ketones seem to be much more stable. So what if we divide the glucose by the ketones and see whether or not we could reduce some of the variation and some of the anxiety that these people would have. So we then replotted Trudy's 2.5-year data using a GKI because we had the data from that one individual. And we replotted it, and everything was so smooth. It was really beautiful.
Hmm.
It was just, oh man. And then I told Trudy, I said, plot your GKI and you won't get so
Worried about
anxiety attack as the result of your blood sugar elevating. And therefore, when you do the ratio, it doesn't look so bad.
Yeah.
And she did that. And we said, wow, this is, so then we went back through all of our literature on the mouse for all the keto calorie restriction and restricted diets and all these things that we did. And we recalculated the GKI and showed that it was linked to the size of the tumors.
Wow.
So you could then predict that the GKI, which now becomes a more meaningful physiological parameter, is directly linked to how fast the tumors grow.
That's amazing.
So if for people to getting into therapeutic ketosis, using the GKI as a quantitative measure that allows people to know that if they're in this zone like you were in, 1.0 or below, those tumors are going to be suffering.
Great.
So you have a biomarker, a readily, easily measurable biomarker to tell a person whether or not they're in therapeutic ketosis.
Right.
And it's better to do it with blood than it was on urine strips. Now, we do urine strips because the ketone strips are a little bit more expensive.
Yeah.
But now they came out with the Keto Mojo, which is a glucose ketone monitor like the Precision Extra.
Right.
But the strips are much cheaper. They're about $1.05 rather than $2.60.
Absolutely.
So, so people can use that as well. We use chemical analysis and we do all these other things, but we did that for years. But we're using strips now too. Mm-hmm. But the whole process now is that you have a biomarker. The problem with the cancer patients, and this is, I get so many emails from all these cancer patients. Oh, I've done everything. I can't get into therapeutic ketosis. My GKI is still around 5 to 8 or whatever. And I said, well, I'm on this kind of drug and that kind of drug. And I said, well, when we did all this stuff with Trudy and the mice, when these people, the mice they were on any drugs. They were not having no drugs. I says, what are you doing? Well, we're taking dexamethasone for, you got to get off all that stuff. Right. All right. You got to what are you taking poisonous chemicals that are going to give you anxiety and increase systemic inflammation? I mean, you're actually interfering with the beauty of metabolic therapy. So I think that, once, and people with cancer have anxiety just because they have a diagnosis of a disease that's potentially life-threatening.
Right. And
that itself elevates glucocorticoids, which then elevate glucose. So it's hard to get into a therapeutic ketosis if you're all stressed out all the time.
Right. Right.
So that's why in the Press Pulse, we put in stress management as one of the key components in the press therapy.
That's right.
And that could be anything. I mean, it could be exercise, it could be yoga, it could be massage therapy, I don't know, what are the needles that you put in your body, those kinds of things.
Acupuncture, right, for sure.
Yeah, that kind of stuff. Right. You can do whatever you think is going to do to reduce anxiety.
And so you're saying reduce anxiety, but what we're really talking about is reducing glucose as a fuel, because you're taking down whether it's steroids or stress or anxiety.
Yeah. Yes, absolutely. So by reducing anxiety, you reduce glucose.
Right.
So if you can get yourself into a new state, a new state of metabolic homeostasis, then these metabolic therapies will really be powerful.
Right.
In maintaining the health of your body, and also continuing to put metabolic pressure on the tumor cells, and also cleans up the microenvironment. So the inflammation in the microenvironment goes way down, just like we showed.
Right.
And you can do all the molecular signaling cascades that you like, but the purpose is to reduce systemic and local inflammation.
Right.
This is going to heal the microenvironment. It's a healing kind of a thing. Mm-hmm. So the ketones are participatory in the healing process of the normal cells. So altogether, you put the whole process together, and it's a really nice, easy. I wouldn't say it's easy. I take that back. It requires a lot of participation on the part of the patient.
Mm-hmm.
If the patient doesn't buy onto this, it's going to be very, very hard.
Right.
If the family doesn't buy onto this, it's going to be hard, because we've seen this happen. So it has to be a whole group participation in the healing process.
And I think the other thing that you brought out in your former talks and so on is that it has to be a gradual process, not just about, the eroding away the tumor, but gradual, I think, for patient understanding and implementation so they don't get overwhelmed with, oh, we got another variable here and here's what we'd like to do. Because in the end, it's actually really pretty simple. But to make the transition, they feel like they're in free fall and this is a crazy guy. Whoever the doctor
well, I think you have to. There's other biomarkers that you can use. Some people use these cancer blood markers. They use imaging. You can do imaging analysis, blood marker analysis, PSA analysis. There's different blood markers that also parallel sometimes. As you start entering to therapeutic ketosis, a lot of these other tumor markers start going down. Gotcha. In general. There's some people that say it hasn't gone down, but in general, most people start to have a reduction in these other biomarkers.
Would you say lactose dehydrogenase is it one of them, or is just one of many? I would have thought that would have been kind of key.
Yeah, a lot of people haven't focused on that. Like when we tell our cancer patients before they start, I don't have any cancer patients. I'm not a physician. I can't treat anybody. But I tell my colleagues that are physicians, get the largest complete blood work you can get on the patient before anything is done to that individual. So we need to look at all the electrolytes. We need to look at all the triglycerides, the cholesterol, LDL, HDL. We need to look at every kind of a blood marker that's possibly available in a clinical chemistry lab that you can possibly look at. Mm-hmm. So just get the full parameter of what this, and you find many of these people are already screwed up metabolically when you look at their blood work when they were first diagnosed with before you treat them with chemicals or radiation or surgery. A lot of them have blood work that's not what we would consider really healthy.
Right.
Not always, but in many cases, their triglycerides are high. Their blood sugars are too high. They have no ketones. It's like, and you look at all these different markers together, and you say, okay. And then as you begin to transition them into the state of therapeutic ketosis, the blood work, the blood work becomes pristine. As a matter of fact, Trudy DuPont had her blood work taken a year after starting the ketogenic diet for her brain cancer. And the physician there in Nice, France, looked at her work and she said, I have never seen an LDL, her HDL was the same value as her LDL. It was a unit, because usually LDL is always higher than HDL.
Right.
So she had a 1-to-1 and she had the lowest triglycerides. And this guy says, you got the blood work of a 21-year-old and she was 60 years old. And so clearly, if you do this therapeutic ketogenic diet right, everything in your body becomes, not everything, but you become younger.
Yeah. No, absolutely. There's a big resetting going on that I don't know how you measure it but you can look at the blood markers, but I think something stays with the person for a while.
Oh, yeah. We did that with the mice. We actually did that.
Huh.
Long-term effects. Their whole physiology changed after doing this.
Right. I have no doubt.
They could eat less food and maintain their pre-treatment body weight with less food. So that means their whole physiology was more metabolically efficient.
That's interesting. It's hard to extract lessons that you're learning directly, but some of them are everybody should fast, I don't know, once a month, once a quarter or something to, and then track for the, glucose ketone index and get therapeutic and then move on. I think it's a great reset in the very least. I wanted to go back to Warberg because through you, I've become very fascinated. My dad's name was Otto, and he came from the German stock and so on, same area. But the one way I look at Otto Warburg, tremendous, family history, his Einstein playing violins and his parents, house, and Krebs was one of his students. And it's a who's who of famous people. Yeah. But, how he didn't connect with the ketogenic diet and yet was so focused on seeing the whole fermentation, you say, in essence, I would have said glycolysis, but it's bigger than that, obviously. But, saying, wait a minute, they're producing lactate, what's going on? And recognizing we got a mitochondrial problem here. And speak to that, because that was kind of one of your discoveries too. You had to go back and kind of resurrect this guy. And by the way, I don't know if you know this, but since your book came out in 2004, Cancer as a Metabolic, that tome.
2012.
12, was it?
Yeah.
So maybe it was one of your studies, because the peak interest of, I'm pretty sure, I'll have to recheck what I'm about to say, but I think you're the guy that tipped off the world of Otto Warburg, because the amount of interest that went up after that was precipitous, and it was kind of dormant for 4 or 5 decades, 3 decades.
Yeah. Well, his theory was that cancer arises from damage to the respiratory capacity of the cell in 2 steps. Number one, the respiratory system becomes inefficient, and in step 2, there's an energy compensation for the loss of energy through respiration.
Right.
So it's a 2-step process, and it's that gray zone between going from respiration to fermentation. That's the transition between normalcy and malignancy. And then obviously, once that happens, he felt it was irreversible. I looked very carefully at his data. I saw Linda Nebling had treated children with brain cancer at Case Western using the concepts of Warberg. And I began to think more carefully because I had seen these same kinds of phenomenon in the calorie restriction when we saw glucose go down and ketones go up. Warberg never knew or discussed the ketone issue. He was primarily focused on the underlying cause of the cancer, which he had steadfastly believed that was damage to the respiration. And I think that's what got him in trouble. We now have a paper that'll be coming out in ASN Neuro to show that Warberg was absolutely 100% correct. Mm-hmm. We're showing now for the very first time that there's 2 forms of fermentation. He only knew one form. And that was the gap in not understanding that the mitochondria can also ferment was the missing link in Warburg's theory. So, in fact, our paper is discussing the reevaluation of Warburg's theory in light of our new finding, which is basically that you can ferment amino acids in the mitochondria. And Warburg knew nothing about this. As a matter of fact, almost the entire cancer field, as we speak at this moment, knows nothing about this. So this is not something that everybody knows about.
Okay.
In fact, my colleague Dr. Chernopoulos from Semmelweis University in Hungary is astonished because he's a big mitochondria energy guy, and he was astonished to find that almost no one in the cancer field discusses mitochondrial fermentation, which is a substrate level phosphorylation mechanism inside in the TCA cycle. Right. So you can't respire, but you can certainly blow out tremendous amounts of ATP from a fermentation metabolism. So Warburg knew nothing about this. Right. And because some of the enzymes responsible for that were discovered and looked at more carefully after he died.
Okay.
So, and the early papers that came out were not well recognized or discussed. So he was unaware. There's nothing in his writings that we could find that showed how amino acids could be fermented.
Got it.
And
can I paraphrase for a second, Tom?
Yeah.
It's like, so what he had discovered, I think was his measurement of oxygen consumption versus lactate production as opposed to CO2, right? So
well, actually, he also showed that many cancer cells can take in oxygen equal to that of normal cells, but it wasn't linked to ATP formation. The oxygen consumption was not linked to the formation of ATP because the cells were fermenting. They were making lactic acid.
Right.
So essentially, the Pasteur effect says, from Louis Pasteur, that yeast cells ferment in the absence of oxygen, and as soon as oxygen comes into the environment, they stop fermenting and they start respiring.
Right.
So you can see respiration is associated with an elimination of lactic acid in a yeast cell with normal respiratory capacity.
Right.
Well, the cancer cell, Warburg grew cancer cells in pure oxygen, not 20%, but 100% oxygen, and the cells are still fermenting. So clearly he argued that there must be something wrong with their respiration because there's no way in hell that they should be making lactic acid in 100% oxygen.
Right. Right.
So he then found this was a characteristic of almost all kinds of cancers that he studied. And he came to the conclusion, and the Coreys, who received the Nobel Prize for the Corey cycle, mm-hmm. were able to show in living chickens that had tumors that they were sucking in tremendous amounts of glucose and blowing out tremendous amounts of lactic acid in a living tumor system, directly supporting what Warburg had been finding in his slices of tumors.
Right.
So the problem is the field thought Warburg had clearly shown that that phenomenon, which is the continued production of lactic acid in the presence of oxygen, which has now become called the Warburg effect,
right.
was the effect of damage to the respiration. So when the molecular biologists got a hold of the problem, they just completely gummed up the whole damn thing, looking at all the genes. And then they concluded that it was the genes that were controlling the Warburg effect, not the mitochondria, because the mitochondria were taking in oxygen. And they were looking at meth, and they were saying, yeah, the mitochondria are taking up oxygen and making ATP, which looks like they have normal respiration.
Right.
But then you give them cyanide and they still do the same thing.
Wow. Wow.
There's no way in hell. And they do it under hypoxic environments too. There's no way in hell they can have normal oxidative phosphorylation.
Right.
So Christos and I originally thought the idea, and I had some little plan in my book, Chapter 8. But then I got linked up with Christos, who's the world leader on that reaction in the world. He's the guy who's focused his career on that one reaction. And he wasn't really thinking about cancer when he was studying that enzymatic reaction. Mm-hmm. So I told him to start looking at these tumor cells. He was blown away. So my God, these tumor cells are all fermenting. They're mitochondrial substrate level phosphorylation, which is the same mechanism that's used in glycolysis. I mean, it's a substrate level phosphorylation rather than oxidative phosphorylation.
Right. So the difference is, and that was all new to me, and I find that really fascinating, is that so now you have mitochondria that either it's damaged, it has its defects, so it can't use or doesn't use oxygen for the most part, but it actually can take in glutamine to make it glutamate, and then you measure that by its end product of succinate. Is that how we're doing it?
Well, succinic acid, and that was another key observation that was made by Hosh Kachka, who since has passed away from cancer, if you can believe it. He had his lab in Hawaii, and he wrote some really interesting papers on animal physiology under different environmental conditions. And he published a paper in 1975 describing amino acid fermentation in aquatic animals that were held underwater. You can't do these experiments today.
Right.
But he took seals and dolphins and some sort of turtle and put them on a board and strapped them to a board and held them underwater. Now these are animals that actually can hold their breath for a long period of time. He wasn't taking some guy's dog or cat and holding it on. That's what they did at the University of Illinois years ago. I was there when they did all that stuff. You can't do any of that stuff anymore. But those are fundamental studies in physiology.
For sure.
But what Kokoschka measured in seals and dolphins that were held, and the turtle, when you hold them underwater, he would take their blood and measure the metabolites in their blood. And the two predominant metabolites were lactic acid and succinic acid. So, succinic acid should never be dumped into the bloodstream or anywhere, because it should be fully oxidized in the TCA cycle. It shouldn't be dumped. So, the end product of mitochondrial substrate level phosphorylation is succinic acid. And now we know that succinic acid is an inhibitor of HIF-1 alpha, which allows the cells to ferment. So, the whole thing is, all the parts of the puzzle are coming together now.
Absolutely.
And Otto Warburg was absolutely correct in his general theory that respiration damage was the origin of cancer. But we had a lag of almost 100 years thinking that the Warburg effect was something other than what Warburg said it was.
100 years.
And
absolutely.
now when our new paper comes out, which should be in a couple of weeks or a month, it's already accepted. It's in the proofreading stage now. We shine light on this process, and I think now we've connected the dots. We've found the missing link in Warburg's central theory of cancer as a mitochondrial metabolic disease. Mm-hmm. And you combine that with the findings that reactive oxygen species in these damaged mitochondria cause genomic instability and mutations. And now you begin to realize that the majority of the cancer entire field is on a bedrock of sand. It's misdirected. So, and that's I think one of the major reasons for what we have today is the cancer crisis, right, which is 1,600 people a day are dying from cancer from a fundamental misunderstanding in the nature of what the disease actually is.
No, I hear you. And so I want to tease that out a little bit. What you're saying is, for those who think that cancer is primarily genetically caused because you were born with a mutation or you acquired a mutation by some exposure, it's not necessarily that. It's basically the mitochondria are not genetically damaged, but they do have defects, and it's in their defects that stops the respiration, which is the mitochondria processing the oxygen, and that's it. And is that primary defect the cardiolipin of the
We don't know that, but we have not yet found a tumor that has normal composition of cardiolipin.
Okay.
And the issue, of course, is you're absolutely right. People think, well, this person inherited BRCA1 and now she's
Right.
her gene is causing cancer. It will cause cancer only if she also sustains damage to her respiration.
Mm-hmm.
If she carries that gene and the gene for whatever reason doesn't damage the respiration, then she never gets cancer. So we call that in the field of genetics, we call that penetrance. So is the gene 100% penetrant? And if any cancer gene can be found that's 100% penetrant, then that mutation would be the cause of the disease. But we have no gene that has ever been found in cancer that is 100% penetrant.
And nowhere near that, and nowhere near 100%, right? It's very...
Yeah, well, the closest one is the Li-Fraumeni syndrome, which is the P50 inherited mutation in the P53 gene, which is about 82 or 3% penetrant.
Pretty high.
Which means that about 18% of the people that have that exact mutation never develop cancer. So, but everybody who develops cancer who has a P53 mutation has defective respiration, because the P53 gene encodes for cytochrome C oxidase 2, which is a respiratory issue. So what if the cell is to survive? It must upregulate fermentation.
Okay.
So you damage the respiration, and a knee-jerk reaction, which is the secondary part, which Warberg said. There's two parts. One, whatever damages the respiration, then must be a transition period to a fermentation metabolism. And once the cell enters a fermentation metabolism, it goes back to its default state, which is proliferation, because what's regulating the growth of the cell and the maintenance of the homeostasis is the mitochondria, which no longer works, so the cell falls back. And the cell behaves as if it were using the metabolism that existed on the planet before oxygen came into the atmosphere. So these cells are falling back on an ancient program that existed. And during that period of time, which was the alpha period, according to Albert Szent-Györgyi, who won the Nobel Prize for his vitamin C work, he said everything fermented in those days, and everything was rapidly proliferating, and it would proliferate until the nutrients in the environment dissipated, and then the cells would die. So metabolic therapy for cancer is doing nothing more than depriving the cells of their fermentable fuels and killing them.
Right.
So we're just bringing all the concepts of biology and evolution and genetics back into its rightful place when it comes to understanding cancer as a mitochondrial metabolic disease.
Absolutely. Speaking of evolution, did you pick up your press pulse template, we'll say, from your evolutionary friends? I think you said one's a chronic stress.
Understanding Darwinian evolution, as much as people talk about it, very few people really understand it. And Darwinian evolution is nothing more than trying to ascribe, the organization of biological creatures on the planet. People get all upset sometimes. They think it's challenging their religion. No, people should never think about that. The concept of evolution is just an explanation of how organisms formed. It shouldn't challenge anyone's religion at all. But a lot of people forget about evolutionary concepts and introduce teleological viewpoints. Cancer cells have a mind of their own. They have an agenda. They have this, they have that. They don't have any of that. This is nonsense. And you find some of the top research papers in the field using teleological arguments to describe the phenomenon of a disease. No wonder we have 1,600 people a day dying. It's incomprehensible. Yeah. And it's so explainable. The crisis that we have in cancer today is clearly, the reason for it is so clear in my mind. And we will not get out of this rut until people can finally understand what the nature of the disease is that we are dealing with. And then once the major institutions come to realize what's going on here, I think then we're going to see major advances and improvements in the care of these patients.
I have no doubt, because I've done all the reading you're reading for the most part. But being older now, I'm a little more cynical of corporate interests and political maneuverings. I mean, was that on the tip of your tongue in terms of why we're stuck where we are?
Well, yeah, I think it's a very difficult problem. It's not just that, we've had these, the Hippocratic Oath, do no harm, right, which has been stretched to its furthest imagination. And then we have the prayer of Maimonides or whatever it is, this. And if you read the prayer very carefully, it says in the physician's prayer, finances and profit should never ever be considered part of a healing process. Well, obviously that's been thrown aside as well. Mm-hmm. And I think it's the educational process. I mean, these physicians are, by and large, for the most part, are entering a field for the good to help people. I think that's the motivation for the majority of people, not everyone, but I think the majority of people, they want to do good. They want to do good. So you go to an educational institution that's going to train you to be a physician, and you learn the basics. But then when it comes to cancer, you learn all of these toxic ways to treat the disease. You don't learn about the concept that this is a mitochondrial metabolic disease. And then the argument is there, well, we can't train the physicians to do that until we see big clinical trials. I says, what about the basic research that supports the concept that's been massively established by tens of thousands of researchers, indirectly not even understanding what they're doing, but when you reinterpret their data in light of the new information, it all comes down to that. So obviously, if a clinical trial were to be done the correct way, I think you're going to get astonishing results. And the problem is, how can we do that in an environment where we have to treat people with toxic approaches as part of the treatments? So when I discuss this with my friends at the top medical schools, they say, well, we can't do the groups you'd like us to do. We can do the standard of care, which is being done all over the place, and we can do the standard of care with some metabolic approach. But we can't do the metabolic approach without the standard of care. And that's the missing control group. That's the key control group. Because what happens if the patients in that group do better than the other two groups?
Clearly. And what if that particular standard of care had to do with radiation, which basically blows everything out? It gives really.
Yeah, I think you have to. Why are we treating patients with so many cancer patients with radiation? I'm not saying we dump radiation completely, because I think there's certain kinds of tumors where radiation might be the only way to really manage that disease. Okay. And brain cancer is not one of them. Brain cancer, people who have brain tumors should never be irradiated, ever. Okay, I can say that right now, and I have evidence to support what I'm saying.
Okay, and we'll just add, but that's because it releases a lot of glutamine, and the cancer cells suck up the glutamine, and they're the that's the fermentation.
In other words, you sign the death certificate as soon as you start irradiating. Right. Now, for 98 or 99% of the patients, that's what you're doing. You're signing their death certificate by just putting them into a radiation mode, because it's very hard for the brain to recover. And some of these people can die from the radiation toxicity, radiation necrotic damage, as well as the tumor. So you're putting these patients at massive risk by irradiating.
And then on top of that, they'll probably put them on steroids, which will bring up the sugar. So now you're back to the two fuels that will feed the cancer.
Yeah, it's just tragic. It's beyond tragedy. It's heartbreaking. It's just, when you know what the biology of the problem is, and then you see people doing these other kinds of things that are putting all these poor people at tremendous risk for survival, you just feel terrible about it. And it's almost like, well, anyway, but the radiation. So the reason they're radiating is because you want to stop proliferation. In fact, the reason you use toxic chemo, is because you want to stop proliferation. Bottom line, why are the cells proliferating? Well, because they have a gene mutation that's causing their cell cycle to be dysregulated, and therefore they're proliferating. So the argument is that, well, they can't proliferate without energy, because nothing can live without energy. So when you say to yourself, what are they using to proliferate? They're using fermentable fuels. Well, if we take away the fermentable fuels, can we stop their proliferation? Yes, we can stop their proliferation, and we can kill them. So why would you need radiation and chemicals to do the same thing that metabolic therapy would do without the toxic effects? Well, we can't use your metabolic therapy because we haven't had a clinical trial. But if you have a few functional brain cells, you should be able to read the literature and understand the concepts.
So, I hear you.
I hate to put it in such a crude way.
You have to.
But the problem is, we got 1,600 people a day dying. It's a crisis.
I agree.
It's a crisis.
So from my perspective, you're having some successes, and it seems like you're having some great collaboration overseas. So you have in Turkey, and you had the case in Egypt. The one in Turkey went really well until she decided not to do it. And
it's hard. I'm not going to say that this is a panacea, because we're learning that we have a lot of learning to do. We're just at the beginning, at the very beginning of this new approach to managing cancer, and we are in fact stumbling a little bit. everybody has a different physiology. Everybody has different levels of motivation and support and systems in place. And we have to control for a lot of different things in order to get people. The physicians have to be on board with knowing the concepts. The patient needs to know the concepts. The family needs to know the concepts. What might be very effective in this person may be not as effective in another person. So we have to tweak the metabolic, the ketonic state. Some people can enter ketosis very quickly. Other people can't. Right. How do we deal with these people? When the patient is finally in therapeutic ketosis, what is the doses, timing, and scheduling of the drugs that we will now use to treat the disease once the patient is in therapeutic ketosis? So if the patient is not completely in therapeutic ketosis, will the drugs be less effective? What doses should we use? What the timing, the scheduling? All of this stuff has to be worked out. So this is the new era. But it's if you're a physician, then this should be the golden era of your profession. Right. Because for the first time, you're actually going to be managing a person's disease with a variety of approaches that will, so you're going to be, the physician should be the master of knowing his patient and knowing what therapy and in which timing and doses and scheduling will work best on this guy, and another guy he can choose. This is going to be magnificent. Rather than sitting this poor soul down in some chair and infusing him with some toxic chemical, and the guy walking away, coming back, and administering some sort of a drug to prevent him from vomiting, that's not what I call medical practice. Medical practice is when you finally can take the knowledge of the metabolism of the person and kill a disease in that person with a combination of drugs and diets and other procedures, hyperbaric oxygen to replace radiation. So we can do it. I think so that's the future. The future in my mind for cancer is very bright. Because right now we're in darkness. We're in a state of darkness. The number of dead people piles up every single year. Every now and then you get a little blip in the curve, but basically it's always increasing, and it's increasing throughout the world. It's not just the United States.
No, I hear you.
So obviously, if we had something that was truly effective, we would have a drop in the number of dead people. And you can evaluate cancer deaths, age adjusted, all that crap. I look at bodies, the body count. You count just like in Vietnam. You want to know how you count bodies, right? We thought we would win the war counting bodies, but apparently we didn't do that either. But you want to kill the tumor cells before it kills the patients, and you want to do it in a non-toxic, cost-effective way. So the other emerging toxic effect is this financial toxicity, which is in my mind immoral. If you see a person, human nature says if you see someone suffering or in stress and distress, you generally do something to help that guy. If he's in a house that's burning, you run in and grab his ass and you bring it out. You don't say, I'm not going to take you out until you give me $50,000.
Do you see that primarily as an American problem? I mean,
No, I see it as a world problem. It's just that we've exacerbated that problem to a very immoral state. But it happens everywhere. And who's going to afford these immunotherapies? And you can't afford these things. They're not going to be available for the general public. And why immunotherapies are also based on the gene theory. So, mutations in the tumor make our T cells ineffective in killing the tumor cells. So we're going to give you CAR T immunotherapy, or we're going to give you one of these other kinds of immunotherapies. And many patients suffer immensely from these things. Some of these things are causing the tumors to grow faster, causing people to have diabetes, causing all these other. You shouldn't treat anyone with the remote possibility that you can make their disease worse. Right? Why are you doing this? Why are you doing this? It's a simple question. Why are you treating people with therapies that could make their disease worse or in any way harm them?
So it sounds like you're saying, by the way, the weakest link in why we're not thinking, we now collectively in the United States, I'll say, is because our med schools have not gone on, either developed their biochemistry, I know you said, hey, well, there's not enough studies out there, but I think that education you're saying that education has to be at that level so you get the early doc at least in their mind, as opposed to, who knows, if we get advanced training. That's not bad, but it would be better if it was in medical school, at least.
Oh, I think so, because, well, it's a biological disease. Okay, we're cutting people, we're irradiating people, we're giving them toxic drugs. Every one of these treatments poses a medical risk to the patient.
Right.
So therefore, the individual is put into the medical establishment only because the treatments have made them vulnerable to things that they would have not had they been not treated for cancer.
Right.
So if you're doing any kind of a surgical procedure on somebody, that's a medical process. So, you know, our view on metabolic therapy is once you use a non-invasive diagnostic test, a lot of people can be diagnosed with cancer without having to cut or poke them. And say, listen, you've got some sort of a mass, you've got some sort of a lesion, it shows up pretty clear. Now we would like to put you under a 3-month metabolic therapy. Okay. And we'll monitor this in 2 months or a month, every month, and see what's happening. And generally, the large majority of people will see a significant reduction in that mass, if not a complete elimination. Okay. If the mass is not completely eliminated in a 3-month metabolic approach, then we can define its borders much more clearly than we could before. Now that person becomes a candidate for a surgical procedure where we can debulk or remove the mass, and the mass is now well defined. And then we think we can cure the disease, because surgery, if done correctly, can cure many cancers. The problem is if the tumor cells escape into the bloodstream, surgery now becomes it doesn't work. It won't cure the disease because the tumor cells have escaped. So then you use chemicals, and we don't have to use we would use metabolic therapies that would target glucose and glutamine to track down and kill those other tumor cells.
Gotcha.
So again, there's the strategy to manage cancer will be under a very different paradigm than what's currently being done.
In talking to you, I think back about, let's say the time of Wilder or Peterman, the whole idea of the innovation of the ketogenic diet. And I don't mean to make this about the ketogenic diet, but at the Mayo Clinic, you know, they had huge kitchens. I mean, their whole getting the meals together. So all of this there was a lot of work behind all that. And, you know, that's not what we hear anymore. I mean, you're obviously part of that. I mean, we're doing this now, but there's been a big gap. You know, what happened to those, call them medical kitchens? You know, they were.
Yeah. Well, at UPenn, they have one because Kristina Berkovic, who's a friend of mine, works at CHOP, the Children's Center there, and they put up a keto kitchen to train the parents how to manage the seizures in the children, knowing how to cook the right foods, the proportions that they give them, and all these kinds of things. And those keto kitchens for epilepsy are opening up all over the place. Okay, well, you can do the same thing for cancer. I mean, cancer can be managed in the same strategy. The future will be clinics. Like you say, the cancer treatment centers of America are basically, they're very similar to Dana-Farber and MD Anderson. They're doing basically the same thing under a different name. They bring in some nutritionists and say it's different or something. It's very insignificant. They're still using they're still using the same toxic therapies. So the new concept will be a center where people go and they are treated for cancer as a metabolic disease. So they would be trained with the keto kitchens. They would have physicians that would administer the correct drugs that would target specifically glucose and glutamine. And they would be cocktails of these kinds of drugs. They would be hyperbaric oxygen. There would be these, perhaps the ozone. I haven't tested that yet, but there's so many new ways that we can manage this disease using a metabolic approach, which would significantly reduce harm to the patient or to the rest of the body and contribute to the overall improved quality of health. And we saw that in the patient that we had in Egypt, where he was a metabolic mess when he was first diagnosed with glioblastoma. And as we began to treat him, his blood work improved. A lot of the parameters of his state improved while we were treating his brain tumor. So we were, the problem with that which was outrageous, is they still had to irradiate this poor fellow, despite the fact that his tumor was under control by metabolic therapy. And we think he died because they did a pathological evaluation of the brain tissue a month before he died. And most of the damage was from radiation necrosis damage rather than the tumor. So the attending physician was outraged about this whole thing. And you also have to keep these people under evaluation for at least 6 or 8 months, I think. I mean, you can't let them, go outside of your care. Because they get off the metabolic therapy. They're not taking hyperbaric oxygen. They start falling apart on what's needed to maintain the pressure on the tumor. So again, this is why I'm saying we're at a learning curve here, and we're going to have an army of physicians that are going to have to think about this and take their skills and what they know and apply it to the problem. And I think there's so many physicians out there that know a lot about the biology, about the physiology of the body. It's just that that knowledge is being misdirected. It's not being mined. It's not being used to its full potential. And I think so many of these physicians could be doing so much more to help these patients if they treated them as the disease as a metabolic disease rather than as a genetic disease.
I hear you. It seems like the bright spots now are, well, certainly Turkey, you mentioned Egypt, Hungary as well, for sure. And
well,
is it
it's going to happen in the United States too. There are people that I'm speaking to who are outraged by this whole system.
Let's take a step up.
And they want to start something.
Okay.
And then the groups of doctors are getting together, functional docs, functional medicine docs. And I think they're forming groups to challenge the AMA standard of care for cancer.
Okay.
Because the standard of care, if you look at it, has been a failure. And the failure becomes from the fact that we have 1,600 people a day dying from the disease. Right. For them to argue that our standards of care have also cured tens of thousands, if not millions of cancer people who have had cancer, I'm not saying they haven't, but those people, most of those people have paid a serious price for their care. They now have all these other maladies that they never had, but for the fact that they survived the treatments that they were used to treat the disease. So we have new branches of medicine opening called cancer survivor medicine, which now treats patients for emotional disturbances, hormonal imbalances, neuropsychiatric, hormonal, all kinds of things, because they were treated with all these toxic approaches. Right. This is nonsense. We shouldn't have to do this. People should emerge from cancer therapy healthier than before they started it.
Absolutely.
Not worse. No, my cancer is gone, but now I have all these hormonal imbalances. I have depression. I have all these other things. This is nuts. This is crazy. Let me
be a little tedious here, because in the components of the metabolic therapy, how we start that, I just think I think it's wonderful, and I love listening to it. And so we start off with basically getting them on a ketogenic diet, and they get their glucose low enough, their ketones high enough, so then we can add insulin to get their glucose down, perhaps even lower.
Well, insulin have to be careful. Yep. You got to make sure the person is insulin sensitive.
Right.
We have to be careful because we have some genetic backgrounds where there's insulin insensitivity, and sometimes that may not. That's where, again, the physician's knowledge of the patient needs to come in. So everybody you have to look at their physiology before you start doing it. Insulin may be good for some, but not others.
On that issue of the whole insulin sensitivity or resistance, you're right. It is a spectrum, but I think it's an adjustable spectrum that it translates into more time at that stage before they get to go to the next stage.
Yes. We know what protects the tumor cells from a lot of the therapies, and that's their powerful fermentation metabolism. And that protects them against radiation therapy, chemotherapy, and all these other things. So, to get a patient into therapeutic ketosis is taking away a part of their antioxidant capability as well through the pentose phosphate pathway. So we know that, and they also use glutamine as part of to make glutathione. So, because glutamate is one of the amino acids in glutathione, and that's coming from glutamine. So clearly, if you target the glucose and the glutamine, you put those tumor cells at massive risk for death. So, and today there's no one, at least I know of, other than the guy we treated in Egypt was the first attempt, I think, in the clinical situation. I could be wrong, but where we actually designed a therapy that could possibly target glucose and glutamine. The problem is we were using drugs that were very weak glutamine target, like EGCG, the green tea extract, chloroquine, that targets diapherases to some extent inside the TCA cycle, but these are not as strong glutamine inhibitors as what I think could be used, like the drug Don 6 days. So the problem is it's very hard to get Don. It used to be used. It's not used anymore for cancer patients. You'd have to have probably some real justification. But we found that the toxicity of that drug can be significantly reduced when combined with a ketogenic diet. Mm. So I think we're going to reduce toxicity significantly when you use therapeutic ketosis as part of the program.
Absolutely. The woman in Turkey who actually did get, I would say, a clear bill of health, and then for whatever reason decided to discontinue on her own, and it turned. You guys didn't do, or it was not included in no glutamine inhibition there. I mean, that was sort of
Yeah, we didn't hit that. And as I said, I think, yeah, the therapeutic ketosis will do a good job at the very beginning and hold the tumor in check. But I think in time, you're going to be killing a lot of these glucose-dependent cells, and some of the cells are going to start using more and more glutamine. So when they say cancer cells are always so tough, it's because you're not taking away their fermentable fuels.
Right.
So they only appear tough, like, oh, I threw the kitchen sink and this tumor comes back.
Right.
Well, it's coming back because it has an available fuel source.
Right.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paper book via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just investing in your own knowledge, you're also contributing to the future of cancer research. Help us make a difference. Together, we can drive change and save lives.
So I don't think it's going to come back, if at all, if you take away its capability of using fuel.
Yep.
Now, if there's a tumor cell that has some level of respiration, that's still intact, then it's possible that tumor cell could be re-educated to enter back into the society of cells, because it didn't go through this arbitrary threshold of irreversibility.
Right.
As Warberg would say, yeah, there's a point when the genome of the tumor cell is so shot to hell that there's no way you can restructure that cell to become part to recover its normal function.
Yep.
So those cells will ultimately have to be killed. But some of the cells that still have some modicum of respiratory capacity, they may be rescued and could return to the society of cells. And now we're learning, it's strangely enough, there's so many things. They talk about all these oncogenes and driver mutations.
Right.
There's a group over in, I think it's England, where they're finding all these tremendous driver mutations in normal cells, right? So it just goes to show you that all these so-called cancer genes that are supposedly driving are now being found in all these normal cells that has no cancer.
Like skin and so on and so forth. Yeah, absolutely.
And esophagus.
Yeah, esophagus too. Yeah, right. Yeah, amazing. So where does metformin come into that? I mean, I know it's an old drug, glucophage, and so on and so forth. Is that? I mean, my interpretation is, is it a glucagon inhibitor so we can control the blood, or is it something else? I mean, and then I hear, well, it has some sort of anti-cancer property, and I have no handle on it.
No, we tried metformin. I think it's probably okay. Trudy was using it. I don't think it hurt her. It's not something that's going to hurt you. It could have some minor therapeutic effect.
Huh.
It's not going to be the big dog.
Okay.
You're taking metformin, it's not going to resolve your cancer cells for the majority of people, but it's certainly not going to provoke the tumor cells to grow. So, like an immunotherapy could potentially make your cancer grow twice as fast. Right. Well, metformin is not going to do that.
Right.
Okay. So, I mean, that's one thing you can know. But whether it's going to, now, as part of a cocktail with other drugs that also target these other pathways that are all responsible for the fermentation, it might have some synergistic interaction with another drug, and therefore metformin's a therapeutic benefit could be potentially enhanced to some degree. Right. But that's going to take a lot of trial and error kinds of experiments to look at that under the right combination. I hear you. And then that. But I don't. When people say to me metformin, go ahead, take some metformin. I don't feel that it's going to hurt you, and I don't think it's ever going to make your tumor grow faster.
Gotcha.
So that's why that's my position on that.
Got it. I think it was one of those studies you told me to read that you oxaloacetate. You have used oxaloacetate. Was that basically to help with the glutamine in terms of? I mean, I'm not clear where it is, but I think it helps limit the glutamine going to glutamate. Is that it?
Right. Well, it could in an indirect roundabout way.
Okay.
And as a matter of fact, the mechanism by which oxaloacetate may contribute to the management of cancer is also going to be a long, drawn-out process, because it's not one of these direct effects on fermentation.
Okay.
It's an indirect way of doing this. And people thought it might have worked by mechanism of calorie restriction, or possibly targeting a little glutamine, but it may eventually be part of a cocktail like metformin. It's not clear.
Okay.
We got some therapeutic positive benefit from it a little bit, but as you can see, the animals still died.
Right. This was a mouse study. Absolutely right.
Yeah. So it wasn't something that was as dramatic as, say, Dawn.
No.
Or combinations of this. We're working to develop here at BC the best cocktail combination that will be used with potentially hyperbaric oxygen and metabolic therapeutic ketosis, and then see if we can possibly resolve some of these very malignant cancers in mice. And it's no easy task. If anybody tells you it's easy to cancer in mice, they're not using the right cancer.
Okay. So,
so if we can achieve that in the mouse, then I think we'll go right to the clinic, and I think we're going to get the big effects right there.
Okay. In one of the studies, it was actually Miriam had sent me this to give me the layout and dig up some questions, that you would use exogenous ketones sort of to help with the ketone levels. Is that something you still look to help, or is it wasn't that big?
Well, I think that's another thing that could potentially be helpful. The key thing is keeping your blood ketones elevated consistently, and keeping your blood sugars low consistently.
Okay.
Exogenous ketones may be able to do that.
Okay.
So the question is, there's a lot of different kinds of exogenous ketones, and I would think that we would need clinical trials or trials on normal people without any disease, to just go ahead and say, okay, let's see how this product works with the GKI.
Right.
So we would need, someone doing a run of a test for about a couple of weeks on people that may want to do therapeutic ketosis using various products. Right. And then by that time, you put it in Consumer Reports and say, hey, this is the product that works best.
Well, along that line of thinking of trying to get the ketones up, have you guys looked into not just MCTs, but C8 specifically? I know that's not a new idea, but I talked to Stephen Kinane about that too. And
Yes. No, it could be very effective. I mean, as you build these so-called therapeutic ketosis platforms, what are the different types of fats? What are the different types of proteins and carbohydrates that are all going to be part of this new thing? Yeah. This is where the food industry comes in. So the food industry is very interested in this. So they want to build products that if you have cancer, try this. Rather than taking some drug that's going to control your vomiting while you're being poisoned by something, right? Take this product. But I can't support or say anything about that because I haven't tested any of that stuff.
I agree.
So I don't want to make a claim about this product being better than the other product because I don't know. People have to try it for themselves.
That's right.
And see if people are out there wanting to get into therapeutic ketosis and you have a product that will get you in there with less grief or struggle, then by all means do it.
Absolutely. But yeah, you're saying you're open to that variable. If there's something that can help us with that side, absolutely do it, and we'll figure out. Yeah. And it varies per person.
Yes. As I said, this is an emerging field. I don't want people to feel that we have the solution to cancer at hand right now. It's going to be an emerging field, and in time we'll be able to say we did.
That's true. I wanted to, it's only because I've read so much that I feel like I have this ongoing conversation with you. But on the press pulse, the idea of intermittently supplying an acute stressor, and that's the glutamine inhibition, that it's important to know that. Because I think some people are going to hear this and say, well, I got my two things down. I'm going to stop the fermentation and stop the glutamine and stop the glucose, and I am good. No, you'll kill yourself. And it won't be a fun way to go. And the reason that we're doing intermittent for the glutamine is because the body also needs it. Where it's a dicey.
Absolutely. You have, now this again, is where the physician's knowledge must be applied. Because, in burn patients, you give massive doses of glutamine because the immune system, to kill bacteria, your skin protects you from all these bugs, and they'll come in and they'll kill you quicker than anything. Sepsis and all these kinds of things will kill you quick. So the immune system needs to be at its highest when you have burn. And the glutamine is a powerful fuel for the cells of our immune system. So if you are too aggressive in targeting glutamine, you paralyze the very cells that you're going to need to pick up the corpses. So if I have a battlefield and I've got a lot of dead corpses on the battlefield and I don't pick them up, you run your risk of causing some sort of a systemic infection. Right. So you need to have an immune system that's going to be at the ready. So if you go and be too aggressive on targeting glutamine, you affect the gut, you affect the uric acid cycle, you affect the ability of your macrophages, T cells and B cells all to function appropriately. So that's why you have to pulse that, right. And you can't press it. And we're even thinking about giving glutamine inhibitors together with high dose glutamine. So in this way, you'll just target the tumor cells while keeping the rest of the immune system healthy. So again, this is a concept that we're working or thinking about right now. So because the cancer cells really need the glutamine, right. Okay. But if I have a drug that blocks it, and again, it comes down to dosage, timing, and scheduling. Yeah. When do you do this? Right. Do you do it simultaneously before or after the drug targeting? Right. So again, but I think these are problems that are solvable. Yeah. That rather than chasing 100 million genes, right. That you have no idea what they're doing. Right. So these are very solvable problems that people should be very excited about.
Right. Right. So the idea of giving the glutamine inhibitor in the glutamine, you're blocking some and then you're allowing some. So you're not killing the body.
Yeah. Well, first of all, if you have the patient under therapeutic ketosis, yep. Yeah. They're not going to be growing very fast anyway, right. The tumor cells. Right. So if I rescue a few cells with some extra glutamine, but if I'm killing 50% of your population and rescuing 25%, you're still on the advantage of killing them. So you just keep doing that until the population is annihilated. Right. Basically. So it's downgrade the population. It goes up by a slight degree. Okay. Downgrade by a bigger degree. Up by a slight degree. So it's a stair-step effect of yep. eliminating the tumor right. cells, rather than trying to kill them all at once. Right. And running into tumor lysis syndrome, which has been a case of some of the immunotherapies. You got to be careful about people running in and wanting these immunotherapies and then dying from the damn immunotherapy due to tumor lysis
right.
syndrome.
They just overwhelm the system, right? The tumor lysis syndrome. Wow. Excellent. Boy, Tom, you're one of the most exciting people I get to talk to, and you just burn bright. I've seen you present 5 times, and I don't know if you know this, but everybody holds their breath or stop breathing about halfway through your presentations because they don't want to miss the next sentence, because you're so full. You don't turn around and take a break,
No, you have a lot of information to distribute in a very short period of time. No. Yeah. And we teach classes at the university level, and we know that we've got to get everything into a 50-minute block. Otherwise, the eyes fall, the heads fall, and the kids just, they lose focus. Yep. So, and again, if you have a lot of information, a 50-minute block, it's like in my cancer, I have a class on cancer that I teach the whole semester. So we go through every one of these points in incredibly massive detail. Wow. Right down to the experiments that people are doing in different papers. And then in my cancer big biology class, where we have 150 students, I have 3 major lectures. And these kids are not biology majors. They're historians and economists and statisticians and these kinds of people. So they're very attentive, actually.
So are these cancer lectures for non-science majors?
Yes.
In the sense that you're now getting the bigger picture to people who need a little more general terms, whatever. Okay.
Yeah. So you got to be able to communicate these ideas to a population of people that never heard of this stuff before. So. And I give lectures to the physicians too, right? Wow. So they're no different than the kids, right? None of these people have ever heard any of this stuff before. But then the advanced biology students, they will read the direct scientific research papers
and your book
that are published.
Right.
Yeah, I'm not expecting the undergraduates in the big non-science classes, to be reading about P53 mechanisms of action. They just have to know the general concepts and how you put it all together. So it's a different audience there.
So given your excitement in the metabolic therapy concept and the students you're teaching, they must be pretty excited. At least I look back, if I had a teacher like this in college who would sort of set your course about what you expect to do, whether it's going into medicine or going to research. Have you got that sort of response? I mean, that you're
Oh, yeah. Oh, absolutely. A lot of these students that are not science majors
right.
become, a few of them, of course, become super motivated to want to do something.
Right.
Whether it's raising funds for metabolic research or whether it's, I had one student who was an economist who got so angry and upset about this current situation. He said, "What can I do?" So I put him on financial toxicity. He's an economist, right? So he wrote a paper it's actually under review in a major journal on the origin of cancer, the origin of financial toxicity in cancer. And he and the professors in the economics department were blown away by the unscrupulous behavior of these drug industries and what they're doing and how they're taking drugs that were found to have a little bit of benefit for cancer, and the drug was $5 a gram, and they're making it now $10,000 a milligram when they find out this is immoral. These companies are immoral. I mean, what else can you call it? What else can you? There's no other definition.
I agree.
So it's, and you tell these, well, we're trying to help people. No, you're not. You're Scarelliing these. You know Martin Scarelli.
Yep.
I tell you, the guy who made the EpiPen. I mean, the drug companies do the same thing. It's just they don't boast
about it.
They're
doing it with insulin right now. Actually, the cost of insulin has gone up by a lot.
So why are they doing that? To make money, right? It's like I'm saying, I'm going to charge you $50,000 to bring you out of the burning house.
Yeah, I agree.
I mean, it's just terrible.
Absolutely.
I mean, it's no moral conscience on these people.
Well, what sounds exciting though, Tom, is you now have your colony of people at the university, at the college, and that are really. I mean, that's excellent. I mean, it's nice to have people who appreciate what you say, and you have a lot of that.
Well, it goes mostly outside of the university because you're in a biology department where they're studying zebrafish, yeast cells, and things like this. So they're not doing cancer research like we do.
Yep. You remind me, however, of George Cahill. I would love to have known him, but anyway, he ended up moving up to he moved up to Peterborough, New Hampshire, and he would go and he would teach a course for non-science majors and professionals at Dartmouth, and it became the most popular course. And they would come to listen to George Cahill talk about.
Oh yeah. Well, the best thing is when you had Cahill and Veach together in the same room.
That would be.
Oh. Those guys were unbelievable. So, but fortunately, Bud is still going strong down at NIH. But, Cahill and I would sit for hours talking about all these, what they used to do to these patients down there at the Joslin Diabetes Center and how they learned about all this stuff. But you're right. His speaking style, and he was a very soft spoken, but the words came out so powerful. And the way he was able to interpret all these things. I think, unfortunately, he passed away.
Yep.
A few years ago. He had I remember when he was getting his knees replaced, he had both knee replacements, and I put a damper on his mobility. So, but fortunately I was around to have him in the early 2000s here at Boston College and talk to him about all these different things.
It's very exciting. It reminds me of what I've heard about Warberg. Is like, wait, so he had Krebs as a colleague or a student in his lab. He had, he was in contact with Einstein, who, obviously, it did things and got him out of the war. And the contacts that he had and the education of batting these ideas around, which must have been very exciting.
Yeah, I think it is. And when you're entering into a field, the best thing to do, a good thing to do is always at least discuss your ideas with people that have a very a deep understanding of the issues. And if they understand the nuances of these issues, they can correct you. If you're off the mainstream. Right. So Christos Xenopoulos in Hungary is the leader in the field of mitochondrial metabolic fermentation, right? Mitochondrial substrate level phosphorylation. Richard Veach is one of the leaders in the world on ketosis and ketone. He was Hans Krebs's last student. These guys are in the bioenergetics, biochemistry field that have spent their whole life in this field. So when you get to the deeper levels of things, it's good to have people there that can keep the boat on the right track, rather than making claims, absurd claims that have caused massive numbers of people to become confused. There are a lot of so-called heavyweights in the cancer field that have published many papers that have misunderstood some of the fundamental issues. And only because these individuals have such a powerful presence, they cause other people to think like they are, simply to get their papers and their grants funded.
I agree.
But yet they contribute to the confusion that's already existing in the field.
And is that more true than not with molecular biologists as opposed to biochemists? I mean, that there seems to be a propensity of defining things through molecular biology and less about...
Yeah, well, genes can't do anything if they don't have energy.
Yeah, you're right.
So everything is energy.
Yeah.
So it comes down to the genes are facilitators.
Right.
They facilitate the ability of a cell to ferment.
Right.
But they are the effects, not the cause. If I take away the ability, the genes turn off. Why, if I don't need to ferment, why am I keeping these oncogenes on? And that's been proven. You move new mitochondria into the cytoplasm, the oncogenes turn off.
That is amazing. Absolutely.
So once you understand the key issues, then all of this stuff becomes a lot more explainable and understood.
Excellent. Tom, I'm going to let you go now. And I so appreciate the fact that you've been, through Miriam primarily, time to talk. I find it very exciting. I'd love to touch back with you as we follow up some studies another time if you ever fit me in 6 months down the road, because I know it's an evolution. I know it's changing a lot. And it's just so exciting to hear this. It just seems so just outside of immediately applicable. These pieces are coming together.
Yeah, well, we need people that are going to be in the trenches that start to apply this to the patient population.
No doubt.
Because ultimately, that's the final frontier.
Yep.
I mean, if you have a therapy that can reduce the death rate by 50%, and it's doing that, then obviously the concepts are correct. Right. I mean, so and yet we're not doing that. And that's the tragedy as I see it right now. So people like yourself and others that have potentially the possibility of moving this field forward, the ball's going to be put in your court at some point, because we can only go so far.
I agree. Well, it speaks to the collaboration, because speaking of being in the trenches, the problem about being in the trenches is that you're with your patients, well over 8 hours a day, and you're tired 5 days a week. You're lucky if you can come to a conference. An extra article so it goes on and on.
I know. It's very hard, but it has to happen because too many people are dying and suffering needlessly. So somebody has to take a break in what they do and say, okay, I got to retrain, I got to relearn. And it's at so many levels, because it's not just knowing all the concepts and knowing how to apply that. Can you do it legally? Yeah. Can you do it without losing your license?
No, I agree.
Right? So there's a lot of things that need to change.
Right. No, that's exactly where I'm at. I wanted to pivot back to you by saying, so people can help fund your efforts by, I believe it's One Cause, One Cancer.
Well, it's the Foundation for Metabolic Therapies.
Okay.
That's Travis Christofferson's foundation.
Okay.
He wrote the book Tripping Over the Truth.
Absolutely.
Yeah, and he set up a foundation to support metabolic approaches to cancer.
Okay.
So he funds our work.
Okay.
And Boston College, you can fund my work, but it has to be very specifically defined. So if people if people want to make a substantial gift to the research,
right,
they have to specifically define that it has to go here. Otherwise, it gets distributed to other programs. So, and it's small, like mom and pop guys, if they want to give me $25 or $30 or $10, which they do,
right.
It's better to give Travis's organization that kind of money because we have a standing research collaboration or grant through that foundation. And 95% of all the money that goes to Travis's foundation goes directly into the research.
Nice.
So, which is very different from any other organization, any other foundation that's out there, as far as I know. The university, takes very little of it through private foundations.
Yeah.
And you can do the same thing. If a gift came in at $10,000 or $50,000 to the university, yes, the university can support, but it has to be very specifically defined and stated that way.
Okay. So best to go through Travis's. Great guy, by the way. Isn't it nice that he discovered you guys, because you have a relationship in essence, Travis
is a good guy, and he is absolutely on the right track of trying. He's doing it from a different angle. He's doing it from bringing the knowledge that cancer is a metabolic disease to the masses.
Yeah.
And I think the whole thing is going to happen from the grassroots, from the bottom, because the patients want this. They hear about it. They want, why can't I be treated with metabolic therapy? And you go to Dana-Farber, MD Anderson, or Sloan Kettering, and you don't hear any of this.
Yeah.
So what's wrong with this? I mean, who are these guys at Sloan Kettering that think they know what they're talking about when they have all their patients, so many of their patients being maimed and die from their treatments?
Right.
It shouldn't happen that way.
Yeah.
So the patients should, and they're getting organized too. So it's just a matter of organization. And as George, you said, is a tipping point that comes, and everybody's going to want this. So, but we have to walk on the hot rocks for a while.
Yeah, no, I think so. I don't know if you know this, but they're working on a standard of care for reversing diabetes via the, kind of through averted health, not entirely. Yeah. But it's fascinating that a number of, a document's been made, and for a number of people to approve, docs to approve it. But I think the same thing, another avenue towards metabolic therapies could be something similar as well.
Oh, so, absolutely. But you're going to have to break through the massive power of the industry, which is not a small industry.
You're talking to a naturopath. We know about that.
Yeah. Well, naturopaths have been on the cutting edge of the metabolic approach, because they do a little bit more. They do a little bit more than some of the MDs do.
I agree. We're also at the place where most of the baseball bats get thrown.
Yeah. Well, everybody's got to deal with their own issues.
Yeah, I get. You're great. Anyway, to that, thank you very much, Tom.
Okay.
So appreciate it.
Okay, Carl. Nice talking to you now.